السلام عليكم ورحمة الله وبركاته. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. السلام عليكم ورحمة الله وبركاته. I'm just gonna, inshallah ta'ala, keep this mask and we'll be getting ready for أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. بسم الله والحمد لله والصلاة والسلام على رسول الله صلى الله عليه وسلم. I'll be getting ready for the Daphnia week 3 experiment, one that I did quite some time ago. But inshallah, I will be able to prepare it with in time. I realize it's on the screen. Okay, so let's present. Okay, no problem. No. Oh, okay. There should be a button on one of the coordinating lab sites that you click, and it should open your door. Yeah. Um, I just don't know if it's this one because normally it's the one that you're right beside. So is there another lab? Yeah. So it may be that side. It will be at right beside the entrance of the lab, like the lab tech's hallway, like their corridor, their room. No problem. So inshallah ta'ala, before we move further. I'm just gonna redo account for attendance. Give me a moment. So before, or give me just one moment, actually. Before we begin, I just want to say that I'll go through attendance once again. When I call your name, you could say yes or here, and we'll be just to make it as proficient as possible. Go through a few action items or a bit of today's schedule. I'm feeling a bit unwell, but if anyone also needs to, wear a mask or take a breather outside, they can. Another thing was as soon as you can start putting in your start beginning soon your quiz. Sorry, before that. Let's see. Okay. So for attendance, do I have Sumaya Abdullahi? No, okay. Jeffrey Adams here. Alfunke Afu Wati. Jaibat. Did I pronounce your name correctly, by the way? Yeah, okay. Ace Cantos. Fit. Sanya. Sanya, is she here actually? No. Okay. Bilqis. Yes. Javitha. Nabiha. Or Nabiha. Nabiha? No. Um, Yiping. Is Yiping here? Okay. Simone. Don't know if I got Simone. Okay. Shireen. Robert. Yep. Owen, I think I saw. I just want to get Alamina. Okay. And Mahima, I believe I got. Yep. Wasi. Yep. Okay. Dorlin. Jaden. Jaden's here. Yeah. Nia, is she here? Nia. Yep. Yep. Okay. Dinu. I think I also know. Yeah. And Alicia or Elisha. Elisha. Did I pronounce your name? Elisha. Yeah. So let's see. 16. that just came in, okay, in the lab, ready, 10 minutes, I'm, book, and I will just take, telling you that, obviously, telling you that, gathering the meetings in more, lab. If you're done the lab quiz for week 3, I would recommend reviewing your lab manual to see what's next for you in groups. We'll be separating into groups of 3. And I just noticed that's going to make it difficult. But we'll talk about it shortly in due time. Yes. Okay, so just want to confirm, has everyone completed the lab quiz? Anyone in the midst of finishing the lab quiz right now? Let's go ahead for timing. I know some of you also may have an assessment or a term test soon upcoming for chemistry. So we'll try to finish in a reasonable amount of time, hopefully making sure that you complete the not only the post-lab assignment, but the entirety of today's lab in due timing. Um, I'm just gonna say, Olafunke, if you can try to pay attention while doing the lab quiz, that'd be amazing. Um, we'll continue for timing. but you know what likely to do to place your things into the cabinet, into the coat rack, any belongings that you have. And if you need a laptop, let me know. But for the lab quiz. Otherwise, you can use your phone. That's okay. I don't mind. In terms of though, so that's the lab quiz, pre-lab number one, attendance done as well. The other thing though, let me just make a note. Um, so I've marked your post-lab assignments, essentially your lab 1 and 2 assignments, and I could provide some feedback. Generally speaking, it's important to be as specific as possible when it comes to mentioning your research questions, your research hypotheses, and being as detailed as possible in following the steps for each question that you're asked. Reason being is because I get a rubric, so I know what exactly I need to look for, but I just want to also bear in mind that you're able to complete with sufficiency or proficiency all details that are required of you. And if you need to take photos, just let me know. Ideally, you'll be able to, for your for the Daphnia experiment, you'll be actually recording your own data individually or in your pairs slash trios. So I'll talk about that in due time. But any questions in the meantime? Any questions? Questions? And as a kind reminder, please do not use your phone when I am speaking. Of course, if you're doing the lab quiz, that's separate, but otherwise, please bear in mind that you're paying attention. FLR. FLR, likely you know by now, if you've looked on Quercus in the module section for our practical 39, you'll be able to see and get access to the shoot biomass and lengths. Bear in mind the units for your figure table caption, right? This is going to be above your table, but also for other important details when you're calculating your mean for the different conditions, control treatment 1 and treatment 2. Please download. Oh yes, and FLR is due on the 3rd of March at noon. So I forgot to mention this as well. Some of you may not know, but the writing assignment number 1 was also due at noon. So bear in mind that the timings are also key because I noticed some of you may actually think it'll be 11:59 p.m. when instead it's actually 11:59 or 12 p.m. indeed. So the idea is that you just want to bear in mind, please keep close attention to details on Quercus for the requirements in the rubric, but also the deadline for your assignments moving forward. And if there's questions, feel free to ask in lieu of the upcoming formal lab report. Academic offenses. So this is something that I'm personally keen on, even though it's mentioned here on the slides from the AO2 practical deck. But the idea is that definitely I expect you guys, as you work in peers or as you work to complete the formal lab report and even just any upcoming assignments for your post labs as well, please bear in mind that you do them individually, such that yes, if you use like citations for references when needed, that's sound. But otherwise, if I see like two people having very similar results, it makes it questionable. And indeed, you don't want to have the risk of, having plagiarism or even unauthorized aid in terms of, seeing other people's results. Also, in terms of concocting, making up different references is definitely something you want to steer away from. And self-plagiarism, if you have questions regarding this, let me know, or you can ask the BioHelp TAs, which should be in a few slides from now. And definitely, I don't want anyone getting a 0 on their FLR. So please bear in mind, especially like don't use AI because I can tell. And I don't want anyone losing marks or get a 0 in any other assignments. Yeah, so I mean, if you have questions, let me know. And I definitely agree. Finish your work ahead of schedule. Try to think a bit about your FLR now. I said 2 weeks ago you could start preparing for your introduction. And in terms of other details, now that you have, the data sheet on Quercus, you can start prepping up for your results. So, and then thereafter the discussion. So just think about the different, details that are asked of you on Quercus, and see how you can best prepare for them in advance, so that way you don't need to ask for other people to support you outside of like the outside of myself, or even just like rather than asking your peers for their files or their data, to rather instead make it like to do it to the best of your ability. So just bear that in mind, everyone. Yes, so Andrea, she's one of the BioHelp Lab TAs, and so if you definitely have questions, she also hosts a milieu of different help sessions. So have a look at them. If you haven't, you can likely also see them on Quirkus previous recordings. So have a look at those as well. And you can find her email on the course syllabus. Now, 2 weeks from now, this is a bit exciting. You're going to go to Toronto Zoo. I haven't been to Toronto Zoo for a few years now, so things probably changed. But nonetheless, for context, you'll be having to arrive at Toronto Zoo before or at 10 a.m. So just keep this in mind independently. Like I was asking if we'd be taking a bus, but actually we're not. So the idea is that everyone is expected to go 2 weeks from now. So this is following reading week to Toronto Zoo as your lab four essentially. So bear that in mind. You can take photos. If you have questions, let me know. We'll be there at 10. I think it's a bit different from yeah, because ours is on Wednesday. You're expected to be there before 10, is my recommendation, because you don't want to be left out, especially as we, get parking passes, and essentially move forward with the day. Um, yeah, so please bear that in mind. Any questions from any of the previous slides? Yes. Um, on Wednesday, like from 8 to 10, for example. And like a practical, or okay, like with that in mind, it's a great question. I would say email biolife.utsc@utoronto.ca, because that's a good question and concern. And it would be difficult to actually go from here all the way to the zoo. So ask them to see if they may likely ask you about alternative dates or timings that you will be available. When you get that confirmation, though, please email me so I can keep note of it, so that way you're not considered absent. Um, and then hopefully you'll be able to have either that TA or myself give you an extension for the lab 4 assignment. Yes. After reading week. The first week, if I'm not mistaken, after reading week is when we're gonna go to the zoo. So you get you get an extra feel of not only biology in real life, but you get a break from the lab itself here at UTSC. So what I'm just saying is that a week, so 2 weeks from now, actually, is it 2 weeks? I think if it's not 2 weeks, it's 3 weeks. Let me just see. Um, yeah, it's 3 weeks. But nonetheless, I would just say have a look at I have the schedule as well. See. So you break for 17th. Yeah, 3 weeks from now. So 3 weeks from now, you'll be going to the zoo. Yes. 20, it would technically be. Today's, yeah, 25. Any other questions? Yes. If you can be there 9:30, that would be great. Technically, if I'm not mistaken, that's when the zoo gates open. But essentially, try to be there as soon as you can. Please don't delay, because, and like myself, I don't want anyone to be traveling or lagging behind, because it'll make it very difficult. Especially if, I would recommend not only having a schedule in your calendar for the timing, but also checking on like any bus system that you use, be it the TTC or Durham bus or GO train, just to keep those details in mind, so that way you can prep well in advance for the other destination. 2000 Meadowvale Road. Any other questions? No questions? Okay. So let's go straight ahead to a bit more into today's practical lab 3. Now we're into physiology, right? So really we're focusing on a parameter that we often talk about, and that's metabolic rate or metabolism in general, right? So what's metabolism? What's metabolic rate? There's like a standard textbook definition. I just want to get an idea as to what you guys know. That's a great response. Any other ideas? What's metabolism, or can you define metabolic rate? Any other thoughts? Any others? Pretty much, it is essentially the breakdown, but there's even more broadly, it's essentially the reactions that exist, both breaking down, like both the breakdown and synthesis of macro and micromolecules in a system. It could be the human body, it could be in a plant like the winter wheat, it could be in Drosophila melanogaster, which is like the fruit fly, and you name it really. So the reason I bring this up is because there are different conditions in which metabolic rate, i.e. the rate of metabolism, can be affected. That includes the energy demands, as you could see on the slide. It also changes in external temperature. And those really influence not only the metabolic rate, but more so the function of organisms. And specifically, they may exist at or they may have different conditions where they're most optimal. So, like, let's say if we have a look here, right? Ambient temperature, and ambient temperature is the temperature in which an organism finds more suitable or essentially retains its core temperature. You find that at different ambient temperatures, so like in the spectrum, you can have an organism that starts low in oxygen consumption and then scales up. Similarly, the case for metabolic rate, which revolves around oxygen consumption as well. So, in a nutshell, different temperatures for different organisms may influence not only their metabolic rate, but also their consumption of different nutrients and waste products. Now, in Daphnia, which is the water flea, yes, question. Tissue? Yeah, sorry. Yes, you should. If you want to go, you can. No problem. Yes, it's a great question. If you need to use the washroom, if you want to break just to drink water, or if you need to do anything, just let me know. Because I do want to make sure everyone's like safe, whether they're outside or inside the lab. Now, Daphnia, as you can see, it's a small aquatic crustacean. It's planktonic as well. And importantly, as you probably saw in your manual, it's poikilothermic, and that means that the internal temperature aims to mimic the external temperature, similar to an ectotherm, in the sense that, they're aiming to essentially match or uh, their metabolic rate and their core temperature is influenced by the environment. And so that's something that you want to keep in mind when describing Daphnia SPP in short for their subspecies, because we'll be dealing likely with Daphnia with its genus name, but the different species, there are different species that exist for water fleas. Let's see. I just want to see timing. I think, okay, I think we're okay. Uh, okay, so let's see what we need to do today. And I'll separate you now into groups. Um, so initially it's saying two, do it in pairs. Let's go with pairs then, essentially to make things easy. Um, I'm going to divide accordingly into groups. Uh, we have 16 or 17 people here. Uh, four, okay, so we have an uneven split. So we'll have a group of three. That's okay. What you can do in the meantime is split accordingly in pairs, two of you, the two of you. I'm gonna bring you close in a moment. The two of you, if possible, can you join her right here? Yeah, is that okay? Either of you could come to each other, but if you come this way, then you'll be able to see the slides more closely as well. Now, in this side, the two of you, Simone and I forgot your name. Nia, Simone and Nia, you'll be together. Oh, now it's gonna be tricky. I know Jay and Owen, one person. Okay, two of you. And it's a bit fun for me. Okay, but we'll say this. Lokis and Yiping. Did I say it right? Yes. And then Robert, Devano, okay. And then Dino. The two of you could be together. If anyone feels uncomfortable as well in the pairs, let me know. But I'll try to briefly run down with you what you need to do. Pretty much, and if you guys want to have a look at the slide, I'll make it easier. So as you can see, there's beakers. These are pipette tips. These are essentially the sticks that you'll be using. As you see it in the lab manual, please keep close attention because you're dealing with organisms. You want to make sure that you don't kill them, you don't harm them, and you do your best to take care of them, especially when you're doing experiments, right? This is in the case of, your BIOA02 practicals, but even down the road in grad school, med school, wherever you may be. So just bear that in mind. You're treating these organisms with due diligence. They're not even our own, right? So just bear that in mind, that they have rights as well. So now with regards to the video, whoa, okay, let's go back. So you're gonna use a small beaker to essentially, okay, you have a big beaker. Big beaker is where you're gonna be taking in large amounts of water. I would say start with just 100 mils or even 50 mils should suffice because the idea is that you'll be starting with different temperatures for further analysis. What I mean is that you have water baths all around that are with 55 degrees at 55 degrees with flasks inside. And then hopefully on your benches, you have ice. You also have ambient water. I'm trying to see if you have extra beakers. Anyone see extra beakers? Yeah. So you have extra beakers here, right, on different blocks around the room. The idea is that if you, I just realized, yeah, perfect. That's good. So just for context, you have a small, or I think it's this one, yeah. You have this plate or this small dish where you're actually going to be adding in a small amount of Vaseline to which you're going to be adding in the Daphnia. Daphnia, I'll help cook for you, but you just want to make sure you add a small smear of Vaseline so that way the Daphnia can adhere into the Vaseline. So bear that in mind, just a small amount. You're just blemishing a small region, probably the size of the navel. So bear that in mind. The larger beaker is where you're first going to start taking recordings of the temperature because you're going to be taking essentially the metabolic rate of Daphnia, essentially the heart rate, across different temperatures, and thereby you need to make sure that you keep track of temperature in your heart rate. So in here, it's around 44 degrees. And essentially, you want to keep in mind that as you adjust or as you add in water and then add in drops of it into that smaller list, that you're keeping in mind that the Daphnia is still suspended, not too much in water, just a few drops, which is shown here, with a small pipette like that. And then essentially, use this other beaker to add in ice, to add in hot water, to thereby essentially finalize the temperature for your Daphnia. Once again, you're adding in only a few drops when you're doing this analysis into the beaker with Daphnia, but you're adding in uh, the right, you're adding in the water at the right temperature. So just bear that in mind. Also, this is glass, right? So you want to make sure that it's not like on the ground or on the side of your bench, but just keep it suspended into the large beaker. And you don't want to run a risk of falling and breaking. And essentially, you have the tools that you need. I'm just going to briefly go through the rest of this or skip a few things, but I just wanted to show it to you more directly. That's the thermometers. Those are the sticks that you're going to be adding in Vaseline to blemish the region. That's the dish where you're going to add in Daphnia or and then Vaseline, and that's the Daphnia themselves. They're swimming, and we have a small amount. That's the Vaseline. So just bear in mind that you're keeping, these details in mind. That's your ice bucket. And that's the temperature, which for us is at 55 degrees, because we're going to be essentially modifying those temperatures. You have a compound light microscope, one of your peers are going to essentially modify it for you, so that way you can have a look at the Daphnia under the microscope. You could use it at the core light that's shown there. And then when you're ready, you can use a clicker to start counting the heart rate. I'll help you see where the heart is, and it's actually nice. The diagram shows it here as well of Daphnia. Um, the heart is essentially around here. If you have trouble, let me know. I think there's a separate video that will show this as well. But essentially, you'll add in the Daphnia inside with the Vaseline onto a small thin smear of Vaseline into that Petri dish as shown. And then you just want to bear in mind that don't add too much nor too little. So really, the amounts that she's adding right now, the lab tech, is a good amount. It's not too much, but it's thin enough for the Daphnia to stick on when you add in a few drops of water to adjust and change the external temperature, in which the polar kilotherm will essentially also adjust its heart rate. So bear that in mind as it adjusts its internal temperature. We'll add in essentially, it'll make impact into the heart rate. Here, essentially, I'll be taking out a fine amount of Daphnia. So hopefully that way you don't run the risk of, the goal is to really keep in mind that the Daphnia are able to live and essentially successfully be analyzed. Um, and essentially, when your group is done that, or even one of the pair of your peers are essentially ready, then you could divide accordingly. Uh, any questions? Any questions? Questions? You can begin. Yes. So that's a great question. There is a setup for the different temperatures. I'll show it shortly, or it may be shown in a few slides. But we're dealing with 5 different temperatures, if not 6. They include room temperature analysis at 10 degrees, 15, all the way to 35. So that I believe is 6 or 7 temperatures, including room temperature. So the idea is that within that setup, there is a certain number of times that you're going to be taking the recordings, right? You know that it's going to be 90 seconds. You count essentially every 10 seconds, essentially 20 seconds. Oh, it's similar. Yeah. Essentially 10 seconds. Every 10 seconds, or for 10 seconds, you'll be taking the recording, like with the counter, if you count the number of beats that you see from the heart. And then you'll take a 10-second break, and you'll do that 9 times to essentially reach 3 minutes per temperature. In total, it should be a good amount. It should be enough time to complete the entire lab within an hour. But I just want to make sure that you're able to, do it with ease, and have another member essentially help record the data in their lab notebook or lab manual, and then you'll be able to upload them for your assignment. Um, Yeah, so any other questions? Any other questions? Uh, in your group, actually, if the three of you can work together, that'd be amazing. Three of you. So let's see if there's anything else. In the meantime, you could turn on your light microscope, whichever one that you're planning to use in your pair or trio. Um, if you want as well to work in trios, that's in my sight more better because it can really help separate the tasks more evenly and you could take time separating, taking turns. But that's your Daphnia now under the microscope. So what you need is one member to start the microscope, another member start taking their a stick with a piece of Vaseline and essentially smearing just a thin layer on their Petri dish. When your group is done, with the thin smear, I'll add in the Daphnia. And yes, you can wash your hands. If you want to wear gloves, you can as well. Trios or duos? The three of you are all. Are you guys ready? So this is an example of how to use the gloves. Some people even use their fingers. Okay. But I only use the glove. Um, so this grid line, we have a decent size, and then when your group is ready, I could start adding in that. Just have one member of your group come by with the Vaseline, with your petri dish, and then I'll take care of hopefully the rest. So also bear in mind that when you're done with the toothpicks, you can place them right here. Want to make sure that we're not touching the Vaseline itself. With the pipette tips, that we have to be very careful so that we don't go in there. If you want to do our best to do that. Okay, so do that. Moving also. You? Yeah. But and then because both of you were the or we could have both of you. If, if you want to work with another like member of another group, that's cool.
I thought that we were before.
Oh no,
Okay.
just two.
Okay, thank you.
No problem. That's good. That's good. Is ready, feel free to line up. Another member of your group can start preparing for the other parts of today's experiment with the water beakers, adding the right amount of water with the temperature of water. You can take it. Yeah. And then the rest of it is on that. We need to put it on. Some of these Daphnia are big. It's even difficult for the pipette to get them. If you want to you can have a careful. How the range of atoms that you- When your group is ready, make sure that you have all apparatuses on. Make sure that you have a beaker with water. So how's everyone doing so far? Testing 1, 2, 3, practical 39. Hope you're all doing fine. Want to ask everyone, how is it doing so far? When you're adding in water, please bear in mind you're adding in small droplets amount, small amounts, droplets in size for your Daphnia from your large beaker. So you have, hopefully you all have room temperature aged water, right, on your benches. So just take around, I would say 20 to 30 mils into that beaker. It's underneath the 50 half mark, essentially around 25 should be enough. And then because essentially what you're doing is adding in more droplets of water. So actually on that beaker, once it has 25 to 30 mils, put your thermometer in, record the temperature that you are on. So that way you add in a few drops thereafter of your Daphnia, of water into the Daphnia Petri dish. Bear in mind as well, you have to be sensitive with these Daphnia because, as I mentioned earlier, they are different species, meaning that if you make a mistake, you have to start from the very beginning. So please bear in mind that if you have questions, if you need my help, just let me know. But don't do things in a rush or without due diligence. So bear that in mind, everyone. There's also counters here for heart rates. I'll answer your question in a moment. What's it? Okay. Are you sure? Okay. Everyone as well. How many of you are having difficulty checking the heart for Daphnia? Is that also Is it related to your question as well? Okay, so let's see if I can show. Okay. So hopefully you all have a similar diagram. You have a diagram actually in your notebook, your lab manual. Please have a look at page 48. You will see the heart opening and closing as an indication of course, in the compound microscope, but it'll be a good indicator. So essentially, right behind this vessel, or essentially the gut itself, you'll have the heart opening and closing. So just bear that in mind. If you need help, it may be the case that how your Daphnia is situated, you may have to tilt it gently to the other side to see the heart more visibly. You don't have to see the entire heart, but only half of it or 2/3 should be enough for you to see it pumping out and in, or up and down, essentially. Your question is.
You have to set the temperature down to 10 degrees and then warm it up, right? Now, I'm aware how to lower the temperature of the water, but how do we increase the temperature? Water bath. Okay. I don't know. I didn't think of that. Thank you so much.
Okay, it's okay.
Okay, thank you so much.
No problem. So just to clarify for everyone, please pay attention. I hope you're all paying attention. Now, one of your members could focus on finding the heart of the Daphnia, while another can start focusing on adjusting the temperature after they get it at room temperature. The idea is for that second individual, hopefully everyone's paying attention, when you are trying to adjust temperature, you have ice buckets to reach at 10 degrees. So make sure you have the second, the large beaker with you, so that way you're able to essentially adjust the temperature and see the thermostat to see if it reaches 10 degrees as you add in small pieces of ice into your beaker. And then once you're at 10 degrees and you've added in drops to measure essentially the temperature at 3 minutes, you should hopefully be able to repeat the process, increasing in temperature using the water bath. Please make sure that you pay attention, because adjusting the temperature is key for this experiment, but also making sure that you do it correctly is as well. I don't want anyone having to go back, and hopefully no one especially breaks the mercury thermostat as well as kills the Daphnia. So if you need help, just let me know. And work with your team members to do things very gently and diligently. Thanks, everyone. Did someone take the sticks? There is a small canister, a small beaker with sticks. Did anyone take them from here, the middle bench? I need to ask if anyone has taken sticks from here, because they are supposed to be staged over here. You can continue on. Just once again, please make sure all materials are placed safely on your benches. The mercury thermostat, you don't want that breaking, because at all.
I see the total down.
I think she
Exactly what it is.
If you're having difficulty, I should help you guys have a look. The heart should be situated on the opposite side behind essentially what you think is the neck of the Daphnia. Essentially, if you see the slide, it's one of the most posterior locations of the body. One moment, I'll try to do it. Yes.
Yeah, and it's really close. Yeah.
Okay. I will open it. So here's a video of Daphnia. So yes.
Yeah, and you will see more clear.
So this is what you're supposed to see, the Daphnia. And I saw one of the groups has a really good visualization. If you need help as well, let me know. But use the contrast disc, that black disc, to help see with the full light feature of your microscope, your Daphnia. So you can start seeing. Keep track of the heart rate as you begin counting. There. What's your question?
If the temperature of the water in the large beaker but once you act on a fish, if they were
That's a good question. If you have additional water that's previously found on this on the petri dish with Daphnia, you could either remove it first with the pipette, the original water, small amounts, because you're already starting off with small amounts of water. So just bear in mind that when you're taking out, you take out first, then you put in the corrected water at the right temperature. That will help as well to make it easier. Yeah. See the brown. Guys. I'll talk about that in due time. There's a list of students asked questions. So, in terms of questions regarding temperature, also. So, remember, you're using the down. So try to keep track at the large meter, the amount of ice that you simultaneously add to see the change, such that the temperature reaches 10 degrees of ice cold water. Then you add in from the water bath, small amounts to increase that the volume and temperature from 10 to 15, from 15 to 20, 20 to 25, 30 to 35, and essentially that's where you'll end. But bear in mind, typical heart rate ranges from 20 to 60 per every 10 seconds. So if you're getting an average amount that range, then that's okay. 20 to 50 every 10 seconds. So that should be okay. If you're getting too low or too high, let me know. You may need another Daphnia, especially if you're starting off at room temperature, if it's too low or if it's too high. If you're unable to see it well, I can talk. Some of them are actually two groups. Basically, remember, you're adding in drops of the ice cold water or 10 degrees water. You're not adding the ice directly to the Daphnia. You can let me know what your observations are, and then I can have it. Or what will be. Now, the answer is right on the previous slide. Admit to the using the first Daphnia, but make sure. So how's everyone doing so far? I want to ask in terms of recording data. Remember, I'll essentially want to account for the different changes in time or temperature you're dealing with. So how many of you are on your second measurement? Actually, I'll ask the numbers that you're on. Wase and Ko, how many, what temperature are you on right now? 10? Your group? 10? 10? Your group? 10? All of you are on 10? Yeah. 10? Okay, so that's good. Just bear in mind as it's important, as you can see what Owen's doing, he's adding in water from the 55 degrees water bath. You add in small amounts, essentially. You don't have to overdo it in your large beaker, nor in your small beaker, because you're only going to add in aliquots, small drops, into the Daphnia. Before you do that, make sure that the water is removed from the existing pool in your dish. And that way you can add in new adjusted in temperature water from your large beakers that have been adjusted to at 15, 20, 25, and 30, as well as 35 degrees. So I want to ask if we're able to finish in about 30 minutes all of the measurements. Because each should take roughly 3 minutes to progress to. So I'll give you about 4-ish or 4.5 minutes. If you're able to finish the remaining temperature, even if you started essentially bear in mind that you want to finish this as soon as you can, and then we could end off with additional observations and measurements for your Q10 data. Yeah. Just before I move further, also in terms of helpful hints, as you can see on the slide, keep the thermometer in the beaker as you're adding in small amounts of ice or 55 degrees water, so that way you could see in real time the change in temperature, and then add in droplets into or on your submerged Daphnia. Try to also keep in mind that the water is on top of your Daphnia, so that way it's like you have the Vaseline, Daphnia, then water on top. So that way you're able to make sure that the Daphnia doesn't regularly move away from where you're observing in the microscope. And then just try to coordinate as members in your group. You could take turns, but try to be as consistent in completing this with efficiency. Now, let's see. Just for timing, everyone, make sure that you're able to complete each step with your partner every 5 minutes. The sooner you do this, the better, and that way you don't run the risk of wasting time. because we want to make sure that we're done at least all 5 or as many temperatures as possible by 12:05. So that's 25 minutes from now. Yeah. Yes. Just make sure you add in small amounts. Oh, so you don't have to use your beak. So this, I would recommend, if you want also, you can have a few more bites, and actually help, but, how are we doing so far in timing? I've heard some of you are at 20 degrees. Nia, how many recordings do you have so far? 15 now? Okay. So just I would say keep it track of time, make sure your group's fully attentive, and hopefully you'll be able to complete as much as possible in the next 20-ish minutes. Or
Kind of reception.
Also, don't forget that when you're done, one temperature, try to essentially remove the residual water without moving the Daphnia with pipette. So that way you are able to essentially do the experiment with the new temperature. At 10 degrees, once you're done, you flush out the 10-degree water with the pipette, place it in a small beaker, and then you essentially repeat the process. What do you think? What do you think? What do you think? Decreases? So, like, have a look at what your results show and think about, actually, even before you see results, why would it increase, for example? Why would it decrease? Do you recall the slide that I showed earlier? The first slide? This one right here? Metabolic rate has a relationship with heart rate. Think about it. No problem. So just to make it easier, this is a range that you likely have for your heart rates. Actually, before I even move further, how are we doing in terms of timing? Do we think we'll be done in about 10-15 minutes? Yeah, how many more temperatures do you have to do? Two? Curtis, how many more temperatures are left? Okay. Okay. How many groups have two more temperatures to count? How many groups have three temperatures to count? Okay, okay. So you should be done in about 10 minutes time. And so I would just recommend, don't rush yourself, but try to brace yourself to make sure that you're able to do this proficiently. As well, some of you may be asking, what's the relationship between temperature and heart rate? Have a look at the slide and you should be able to get an idea as to what their relationship could be as you see the relationship between temperature and metabolic rate. So as a recommendation, please go check yourself. I would, like close to 25 hours. Yeah, doing 225? Yeah, we're like, okay. Also, this is every sport like, 60, because we're going into high. No, I mean like not 60 degrees, like 60 weeks. Generally, you likely sloping on. Okay. The question is, though, when you began at 20 degrees, did it go up or no? Like when you added it in, it was at 20, right? Yeah, it was at 20. Before, oh, okay, so, but you still added in to drop that 20, right? Yeah. Because it didn't go as per the second question is, So what's your average?
Average for
20.
20, it was looking like 55, 20,
Continue as you are. I would just say.
and then for some reason, 10 degrees is higher than 15 degrees. Because 10 degrees was like 50, 20. 15 degrees was like
You will be I'll get an idea to getting the general. But hopefully, I would say continue just making sure that you are keeping the right temperature. You don't necessarily even have to use high temperature because it just reaches 25. Which, really, try to also make sure that your beakers don't get filled up to the top because you don't want to also have them spill. So if your small or large beaker is too high in volume, then try to place them into the sink, half of the size, because generally speaking, the temperature, if you mixed it well with the scupula, should be able to maintain its temperature. Also, don't forget the mercury thermometer that you do not leave it on the side of your bench to roll. How's it going for you? Okay. How many more do you have? Right? So at 12:15, just want to confirm actually, moving forward, how many more recordings does your group need? 2 temperatures left? 1 more? Okay. How many more, Simone? 2? Jaya? Done? Okay. How many? 1? Okay. Your group as well, I think your group is how many temperatures? Left. 1 more or 2? Are you at 30 or 35 or 25? You're at 20? Okay, so 3, 3, okay. 4 or 3? What, 1 more? Okay. Wase, how many more does your group have? Okay. Okay, so I'm just gonna say, okay, let's see. I'll wait to see before I say anything else. In about 16 minutes, I'm gonna have to go through the rest of today's practical. So try to finish up the remaining pieces. And remember, by the way, folks, this is a team effort. So I do expect that you're contributing fairly as a group. Each member is supposed to play a particular role, be accounting or looking down at the microscope. Please bear in mind that you're taking tasks fairly, so that way you're most efficiently as well able to complete today's lab. So you have 15 minutes. Some of you have 3 more temperatures to go or 4. So that should be enough time actually. But at the same time, don't try to go straight from one temperature to adjusting for the next, and hopefully you'll get what's best. Let's see. If your group is done, or if one of your members are done, they could start putting it in onto Excel. the results that they have, including all values that you see in your lab manual from the table that you were able to essentially include data. That's table 3-1, page 52. And then you can start reading into page 53. Once your group is done, looking at exercise 3 to begin data interpretation and analysis. I would recommend starting getting ready with this as soon as you can. And then hopefully, if you have questions, let me know as we go forward in about 10-ish minutes, the rest of today's practical. If you are done, I want you to carefully add water. Actually, I'll show you. So, very safely, very safely add in water to one edge. So that way, actually, we'll do it here. And have it at ambient temperature. If you're done, essentially, we could do essentially take care of the waste both ways of your beaker with water and of your Daphnia. I don't want anyone filling their Daphnia at the very end. There's no purpose and no right to do so. So the idea is that when you're done, carefully let the Daphnia stream off, so that way they don't essentially have to remain desiccated or dry off. You use your beaker, you gently rinse it, and then you're pretty much done after cleaning up the remaining bad fleas. Once your Daphnia has left pretty much the teeth. When your group is done, they'll be able to upload the data onto their own Excel sheets, and they'll be useful for the lab assignment. If your group needs help, let me know. Once again, carefully add water for the Daphnia when you're done for cleanup. Don't like harshly add water to the top of the Daphnia. Let them essentially swim off by gently adding in water from one edge away from the petri dish center. Anymore. Yes. Thank you. About -13. If you're running out of time, then I recommend taking 5-second breaks rather than 10 seconds to make sure that you're able to keep the remaining amount of temperature. You can also not have the 10-second break, and you can just continue in tandem to keep recording going as you record. Just take breaks to count and then repeat the process every 10 seconds. And that way you save half the time. Any questions, concerns?
Do we fill it up like this, or do we just put down the exact temperature?
It would be identical.
Oh, the exact temperature?
I see you have 23 degrees.
Yeah.
Is that 25?
23.
Yeah, so you put the exact interval that you had.
So this interval, you said that we can reverse the order, but do we put down 13 here or minus 13?
You put 13.
13.
Okay, one of the people I meant like they tend to how you have it here is cool. But I'm a bit confused. If you want me to show you. Oh, sorry, yes, that makes sense. Because I was looking at these.
Yeah.
That's pretty much what you do. Keep doing it.
Right, so if with this one, we don't even need to think about the switching order in the formula, like, yeah. Okay.
Who- Is anyone? Is anyone? So, team members, I just want to make sure you please let me know if there's anything that's broken equipment-wise. It's very important that you inform me because one, it is our responsibility to make sure that we keep the equipment not only clean, but intact throughout our practicals together. Do not just hide it because if I don't find it, the techs will find it, and that's not nice for them. So just bear that in mind, everyone. Make sure if you have questions, concerns, something's about to break, or you're worried that you may have broken something, just let me know. We'll do our best to help take care of it, and I can inform the techs if needed. But you don't just hide it because that is a deterrent for yourself and your team members, as well as the entire lab and those that use it in following weeks. So please bear that in mind. Please let me know, keep me up to date, and I really appreciate it. Thanks a lot.
Remember the going earlier asked you if we need to switch the temperature because of the temperature interval, but if with this interval, we don't need to do that. We just use the formula.
Yes. So you pretty much for that table, you're showing the solution, like the solution of like
So do you want us to just put down the exact temperature?
I would prefer you even show that we put the range.
Oh,
So you have it. That's, no. You can't actually. I would love to, but the issue with the Vaseline is on the Daphnia. If Daphnia also in that beaker are in contact with Vaseline, it may not be too well for them. So when you're done, just let me know, and I can help you. You're done? Okay. How many groups are done? Please raise your hand. All members of your group can raise your hand. For those that aren't done, please tell me how many more temperatures you have left. One? How many more? One? One? Ace? How many more? One? You're finished? Any other groups here? How many more left? All done? All done? Simone, Nia, are you guys done? Alaykum, any more? Any more? No? Your entire group is done? Yeah. Okay, perfect. So when your group is done, just let me know. We'll be speeding them up fairly. Your group is done. You can begin cleaning up your benches. Make sure once again you have the dishes that are removed from the Daphnia that should be rinsed gently and carefully down to the drain. Also, make sure that the smokes are shut off. You don't have to take out the wires, shut off the light, and make sure that everything else is returned back to where they were before. The sticks, if we use them, we can place them where the disposable sticks are located in the bench. Kim White can be placed into the garbage and paper towels as well. Once your group is done, let me know, and you can start actually paying attention to the remaining slides we have left. Do not pick up your last book as of yet. You're not officially done. because there are a few other important things that we need to complete before you leave today. You can clear up your bench. You could even begin packing up your bags. But in the meantime, make sure that before you leave, you pay attention to the remaining few instructions there.
In the last, that could be going on, but it's okay, so I just. Literally, walking on the kid smashed on a chair, like, you know, when you were walking out. Walking. Yeah. No, but I don't know, some, the chairs couldn't fall, so it was all the way off, so I was moving it.
You can regulate the chair, right? Yeah, the information of the chair, you can regulate how high.
Yeah.
Let's say another piece of glass is already cut when you begin any lab, or you find that it's weaker than what other equipment you use. Just let me know. It's okay.
Like I know, but I don't think like how much block did I get less from?
Is it the
Slideshow one, like, you know, we have to make a presentation on the three slides. Is it, I get like that?
yeah, I can have a look at it. No problem. And this is lab 2. Okay, lab 2. No, but like
Oh,
lab
no,
two.
that was that.
Yes.
Thank you.
No problem. You're done? Okay, so in the meantime, I do want to make sure everyone else is almost done. Are you guys almost done? Almost done? Yeah, almost done. Wase and Cole, you guys are done? Yep, okay. So begin packing up in the meantime, but I want to make sure that you guys can actually start finishing your lab assignment now. If you have your computer with you, you can actually work on it now. I know some of you may have term tests. That's okay if you have to leave, but I just want to make sure that you're able to complete the rest of today's lab with ease. First off, you have the data points. Don't forget to calculate the mean. When you've calculated the mean heart rates from table 3-1 across the different temperatures, right? And you'd round to the first decimal place. When you're done this, you start calculating Q10. Q10 is your measurement to be able to depict how related or how impacted you find heart rate to be by temperature. So this is essentially the formula that you see in your manual when it relates to different temperatures that are separated not by 10 degrees. So if you have 13 degrees and 17 degrees, this is where you calculate your Q10. Or if it's 13 degrees and 20 degrees, then you calculate Q10 in this way. If it's 13 degrees to 23 degrees and there's still a 10 degrees sharp difference, this is the formula instead that you use. And so you could easily deduce your Q10 value. If it's closer to 2, it depicts that there's a strong relationship, essentially correlationally, between your dependent variable with your independent one, heart rate with, in your case, temperature. And if it's closer to 1, then there's less of a relationship. So it's more correlational in this quantitative manner. And then remember, for the last remarks, last 2 slides, before we end off, lab 3, your assignment is shown as on the slide. You download and transfer your data. Remember that you have group data, but you're going to format it uniquely for yourselves. Don't copy and paste from each other, especially because I can tell. And in terms of your table caption, make it as specific as possible and maintain the specificity of details that we've touched upon before. When you're making a caption, be as detailed as possible. What type of organism are you looking at? What temperatures did you explore? And, mentioning these details are key. Creating a 2D line graph is a simple line graph where you could connect, let's see. Yeah, pretty much a line graph on Excel. And then essentially the details are shown here. Mean heart rate, right? Specify the units as well. Number of beats per 10 seconds on the y-axis. Temperatures in degrees Celsius. You can also note other details as shown here, but I've already touched upon them. Just make sure that you're specific in all details that you include on Excel or as a photo that you send with your name and other details as shown before. Your figure caption or your table, your Q10 table, and the mean heart rate will be sent separately, but you need to have them labeled with your captions respectively. Table captions for FLRs go on top. Table top, figures below, so underneath essentially. Think of it like that. Your figure graph captions are always underneath your figure, whereas for tables are above. Lastly, let's see. Please complete. Yeah. Well, not. Yes. So make sure all of your team members have actually copied the data from any peer's lab manual. Make sure that you complete this within 24 hours. If you don't, you're losing 10%. So I don't want anyone losing 10%, but you have to do your best and leave the rest with the best. And that's before I end off. I hope you have an amazing reading week, an amazing one indeed. And then hopefully, lab 4, 3 weeks from now, February 26th, if I'm not mistaken, 10 a.m., we'll go to the zoo. How does that sound, everyone? All good? You're all done? Don't forget to shut off your microscopes. Have an amazing day. If you have any questions, let me know. All the best for your term test 1. Take care, everyone. Bye.
For this table, right? Do I need to keep this, or is this the caption?
It's pretty much like your caption.
Oh.
You can specify more details though, as like specify that you're dealing with 6 intervals and that you're calculating something 10. You can go through, but you would also put the units, so degrees Celsius, and then heart rate, the units that we talked about, which is like beats per.
That's enough. For the
And then the thing I just mentioned about 6 inches.
intervals, yes. And so I see. So I just put it above the table to replace like this part.
Yeah.
Okay.
Within your submission, you would include the table, and then above it, those details that I just mentioned in that table.
Okay, thank you.
No problem. Any questions? Yes, pretty much will be this entire. Okay, no problem. If you have,
I would explain here.
yeah, it's very correlational. Yeah, you got it. The idea is that, like, if do you have like a calculator that could do logs, by the way?
I do.
Yeah. Perfect. Okay, so essentially what you do is, like shown in this example, you have essentially the heart rate, pretty much your mean value with a higher temperature. If it's like, let's say,
Like for like like 20 degree.
yes. If let's say this was 20 degrees and this was 15 degrees, then essentially that's what you would calculate. Like that's, so the R would be the number of heart beats that you have shown here.
If 15, could I do like 10 or?
So you're actually going to do it 10.
Oh,
Yeah. So they're all shown here. So you'll be essentially taking it from 20 to 30, 15 to 25, essentially going in between 10 degrees. Okay. If they're exactly 10 degrees, then you use this formula, the simple division. But if you are, let's say, a few degrees off,
like 9.5.
yeah, and the more specific you are, the better for your results. So it's okay if you use this formula, and also help train you for future use. In that, essentially, the temperature will be in the denominator, right? The range difference, and then essentially the values that you have for your mean heart rates will be in log at the numerator, and then you multiply.
And this is essentially telling us how sensitive the organ is.
dyspnea is the temperature.
It's heart rate.
Heart rate.
Yeah.
So sensitive to heart rate is the temperature.
Exactly. Yeah.
Thank you.
No problem.
We have to calculate the Q10 values with each temperature.
Based on the ranges that are shown in the lab manual, there is like between 15 to 25, 20 to 30, 25 to 35. So there are different ranges. Pretty much I just showed her, and they're also here. So have a look at page 53, and then you'll essentially take note of the different the ranges.
Also, is it the highest?
Yes.
Also, are we gonna be graphing the Q10 values ourselves?
No.
Also, it's just like our regular values that we got.
Yes. But make sure to include the mean, and I recommend people also including the error bars. I don't think they were shown here. It's okay if you just put the mean, but it would be a bonus. Like for me, saying like it would be better to include the ranges because you have a standard deviation that you could account for.
Okay. You know, study positions are available, like for students or anything, like research positions for students are available.
Mainly they're shown.